The information in this module is accurate and complete to the best of our knowledge. All recommendations are made without guarantee on the part of the author or the sponsoring institutions. The author and the sponsoring institutions disclaim any liability in connection with the use of this information.
Welcome to the Population Differences lecture. In this lecture, we'll look at apportionment of human diversity. In other words, how much variation is there between human populations? Humans, despite their worldwide distribution, show very low amount of variation compared to other species, and most of our diversity is still in Africa. For example, no autosomal marker differences between populations have ever been detected to be more than 25%. Well, results from mitochondrial DNA and Y chromosomes are somewhat different, with less of variation within populations and more between groups, as might be expected from the smaller affected population sizes and hence greater genetic drift. So the question is, can we use genome variation to study population differences and assign individuals to populations based on the variation of markers in their genomes? Several statistical techniques have been developed to assess genetic distance and population structure. We'll look at three: multivariate analysis, multidimensional scaling, and cluster analysis. Multivariate analysis allows us to reduce the multiple dimensions to 2 or 3 dimensions that we can draw and comprehend, while minimizing the inevitable loss of information. Multidimensional scaling, or MDS, is one of those methods where the loss of information is represented as stress statistics. With the lower than stress statistics, the better the MDS fit to the data and therefore the loss of information. Finally, cluster analysis is to group individual into K different clusters based on genetic similarity, so that individuals within cluster are more similar to each other than individuals outside the cluster. Multidimensional scaling is means of visualizing the level of similarity of individual cases in a dataset. It asks a question: how many dimensions are needed to display population relationships? Distance matrices are shown here relating genetic distances between increasing number of populations. As the number of population increases, the number of dimensions needed to display these distances visually, such as the lengths of the lines represent genetic distance between them, quantitatively also increases. The researcher must decide on the number of dimensions before analysis. The more dimensions, the better the statistical fit, but it is more difficult to interpret the result. Alternatively, principal component analysis, or PCA, is a mathematical organism that reduces the dimensionality of the data while retaining most of the variation in the dataset. Individual axes, known as principal components or eigenvectors, are extracted sequentially, with each PC being independent and encapsulating as much of the remaining variation as possible. In this example, graphical representation of principal component of analysis of 5 populations in both 2 and 3 dimensions is shown. In this example, the first 5 PCs account for 95% of the variance within the dataset. The first 2 PCs are shown on the x-axis and y-axis respectively, with each individual plotted according to their eigenvalue for the first x-axis and second y-axis principal component. These principal components can be used as axes for graphs that minimize the amount of variation displayed in a fixed number of dimensions. Ancestry informative markers can be used to estimate the geographical origin of the ancestors of the individual, typically by continent of origin, for example, Africa, Asia, or Europe. The question is therefore, how precise an origin can be specified? In a famous example, a rotated PC plot was used to highlight the special similarity describing the first 2 PCs and the geographical distribution of samples in Europe. This paper by John November et al. 2008 was called Genes Mirror Geography within Europe, and was a quite famous paper. Alternatively, cluster analysis or clustering is a task of grouping a set of objects in such a way that objects in the same group, called a cluster, we have clusters A, B, and C on the figure on the right. These are more similar to each other, they're closer to each other than those in other groups or clusters. Genetic markers can be used in cluster analysis of the degree of similarity of genetic data between individuals and groups in order to infer population structures and assign individuals to hypothesized ancestral groups. They can also be represented by a tree. As you can see, cluster A, B, and C represented as a tree. The result of this genetic cluster analysis then is a matrix where each individual is given a value or membership coefficient to ancestral cluster, and these should sum up to 100%. Detecting population structure from genome-wide data by cluster analysis can be performed in computer algorithms. We see here a typical output from the program called destruct. This using data from a program called structure. During each analysis, membership coefficient summing up to 1 was assigned to individuals for each group. So each column is representing one individual. But you can see each individual in fact has different colors assigned to it, which colors represent different groups to which membership coefficient was assigned. For each individual, all of these colors add up to 1, but each individual could consist of colors giving different membership coefficients. Overall coefficient matrix, termed the individual Q matrix, is generated with rows for the number of individuals and columns for number of clusters, denoted by K, or K clusters. The aim of genetic cluster analysis is to group different individuals into K different clusters based on genetic similarity. We can estimate the K before starting the analysis. So the K value, so number of clusters, that gives the best fit to the data, which should be the estimate for the true number of clusters. For example, on the right, you can see increasing K value above 5 gets to a plateau, so it does not contribute meaningfully to the interpretation. But also, you can use K value based on prior hypotheses. For example, if we are analyzing population affected by admixture from one or more other populations. For example, if Puerto Rican population has been made by admixture from 3 different sources, African, European, and Native American, then there should be 3 native clusters in the Puerto Rican population. We can also run the clustering algorithm several times and present different values of K clusters for 1, 2, 3, 4, 5, and 6K, as represented at the bottom graph. While differences between human populations are a small proportion of all variation, cluster analysis allows population assignment. With a large number of polymorphisms, considerable information about population of origin of an individual can be obtained, sometimes down to the level of individual country or even a village. I prepared some review questions. Can you describe methods involved in describing genome variation between populations? Contrast PCA and MDS methods. What is cluster analysis? Explain the ways of choosing the optimal K for the cluster analysis. What is the computer program most often used to describe the structure of human variation? This concludes today's lessons. Population differences.